In chapter 21, we are going to study proteins and amino acids. And we're just going to start with definitions of proteins and amino acids. Protein is a very large biological molecule. And just as a reminder of what it means to be a biological molecule, it means that it is synthesized in the body, or it has some sort of purpose inside the body, or both. A protein is made up of smaller molecules called amino acids, which I'll define in a second. And each protein has a varying number of amino acids, but it's a ton of them. Proteins' jobs are to transport. So they are moving molecules throughout our body. And also, they help regulate our growth, our development, and our immune system. And we don't spend a ton of time talking about proteins in this class because that's more of a topic for biology. We really focus on the synthesis of proteins. What are proteins made of? These amino acids. What are they and how do they come together to make a protein? So now let's talk about the amino acid. The amino acid will define as the building block of a protein. It gets its name, amino acid, because it contains two functional groups. It contains an amine, and it also contains a carboxylic acid. So that's where the name amino acid comes from. There are 20 naturally occurring amino acids. And at no time in this class will you have to memorize all 20 of them, not their names, not their abbreviations, not their structures. But we will be looking at the table of the 20 amino acids. All amino acids have the same general structure. It's very standard. They all have a carbon atom right in the center of the amino acid. They have 4 bonds coming off of this carbon atom. And the way that I'm going to draw the generic structure for your amino acid here is going to look a little different from the way that you'll see it in your textbook, but I'm drawing it in this particular way for a reason that will be helpful later. So one of the things on the carbon atom is a hydrogen. All amino acids have one hydrogen on their carbon. And then what I'm going to draw to the left side is a nitrogen with 2 hydrogens. And on the right side, a carbon with a double bond to an oxygen and then a single bond to an OH. So over here on the left-hand side, the NH2, this is the amino part of the amino acid. And then over here on the right-hand side, the COOH, that's the acid part of the amino acid. And then this last bond that hasn't been filled in yet, we're going to abbreviate R. And this R is the only thing unique about each amino acid. That R varies for each amino acid. And it is how we tell one from the other. One more thing that I want to put in here for notation, you're going to see this come up at different times when you're working on homework. That carbon that's straight in the center of the amino acid is sometimes called the alpha carbon. So just kind of keep that in the back of your head. So this general structure of amino acids is not how they actually exist when they are inside our body. When an amino acid is at what we're going to call physiological pH, physiological meaning the pH that of the inside of a living system. At physiological pH, amino acids are ionized. And this ionization process involves taking this hydrogen atom from the acid portion of the amino acid and actually moving it to the other side of the molecule. So it moves over to the amino part. And what that looks like, it's the carbon, the alpha carbon that still has the hydrogen on it. Now the nitrogen, because that highlighted yellow hydrogen has moved, the nitrogen now has 3 hydrogens on it instead of 2. And over on the acid part of the amino acid, we have a carbon-oxygen double bond and then a bond to an oxygen, but that hydrogen is gone. The R group is still there, whatever it might be. When the hydrogen moves from the acid over to the amino, it takes, it leaves behind its electrons. So this means that, I don't know what just happened there. So this means that for the oxygen is left having a negative formal charge, which I'm going to circle, which is just standard notation for organic chemists. And the nitrogen ends up having a positive charge, which I'm also going to circle. So as a result of the hydrogen moving from the oxygen over to the nitrogen, the amino acid becomes ionized. It becomes a cation/anion. This particular type of ion, something that has both a positive charge and a negative charge all at the same time, it's not a cation, it's not an anion, it's called a Zwitter ion, which is maybe something that you heard in Chem 121, but I feel like probably not. So a Zwitter ion is an ion that has both a positive charge and a negative charge. So let's take a look at a table of amino acids so we can kind of get an idea of what some of these different R groups are. Now I have, you have a table in your textbook. I have a table here that is different from the one that's in your textbook in the way that the amino acids are organized. You can find different tables of amino acids on the internet all over the place. They're everywhere. Find one that organizes the amino acids in a way that makes sense to you, in a way that you like reading. So for me personally, this is the one that I like. And on this particular table, note you'll see that, let me get my pen here. So on this particular table, we have the R groups are kind of shaded in boxes for each amino acid. So that's sort of how you would see them in the structure. Remember, I told you when I was drawing the generic structure of the amino acid, I don't draw it exactly the same way as it's drawn in textbooks and things like that. And specifically, what I draw differently is the position of the COO minus relative to the H, like I swap them. I do that for a very specific reason. Like I said, it's going to become helpful later. The different structures of the amino acids. And this chart also gives us the names of the amino acid. Doesn't give them give us the abbreviation, but it does give us the names. Notice that there's a couple of amino acids that go by 2 different names. So aspartate and aspartic acid, 2 names. You could see it called either one, glutamate or glutamic acid. Another thing about this table that you're probably paying attention to is that I have written on the table things like neutral and acidic and basic, phrases like this. And so what I want to talk about next is what those different things mean. These are ways that we have of categorizing the amino acids. So what we're going to make here is a tree diagram of how you could categorize the amino acids. Now, I like to have a table like that one that I just showed you that already has the amino acids categorized. But if you don't have a table that already has the amino acids categorized, you can use this little flowchart type thing to categorize them for yourself. So the first way we'll distinguish amino acids is by calling them either nonpolar or polar, which are words that you heard and used in Chem 121. For a nonpolar amino acid, the R group that's attached, what you drew in green, what I drew in green, the R group of the amino acid is either hydrogen or it is a larger group that contains only carbons and hydrogens. And a polar amino acid is one with an R group that has something other than carbon and hydrogen, an oxygen, a nitrogen, or a sulfur. So let's go back to that flow chart again and let's look at some of these amino acids. So we can see here the amino acid up here, lysine, this guy right here. If we're just focusing on the R group, there's a nitrogen atom right there, which makes it polar. Or if we look over here at isoleucine, if we look at, again, we're only looking at the R group. If we look at every atom in that R group, all that we see are carbons and hydrogens. So this one is a nonpolar amino acid. Same with leucine. All we see are carbons and hydrogens. That makes it nonpolar. But if we go down here to serine, we can see an oxygen in its R group, which makes it polar. And some of these are in line structure in this particular diagram. Like over here, phenylalanine, all that you see are carbons and hydrogens and nothing else, so that's going to be nonpolar. Okay, so let's go back to our tree diagram. Once we've divided them up as polar or nonpolar, for the nonpolars, that's pretty much where the categorization ends. For polar amino acids, we can divide them up even further. The polar amino acids can either be neutral, and sometimes we'll call that polar neutral, or they can be acidic, or they can be basic. And this is referring to acids and bases, just like you learned about in 121. Acidic polar amino acids have an R group with another carboxylic acid functional group. So the R group on an acidic amino acid is going to have, sometimes it might be written out as, I need to give myself more room here. It could be written out in shortened notation COOH. It might be written out with a charge on it, so it could be COO minus. It could be written out in long expanded notation, so there would be the COOH, or the COO minus in expanded notation would look like that. So you could see any one of those forms. For the basic amino acid, our R group has a nitrogen on it with a positive formal charge, or we can see a nitrogen with a double bond. And again, this would only be in the R group because all amino acids are going to have a nitrogen in them in the amino part, and they'll all have a COO minus in the acid part. All the other polar amino acids are classified as neutral. So let's go back to that chart again and look again. So we can see, like looking here at glutamate, you can see the COO minus group on there, which makes it an acidic amino acid, and that's where that notation is coming from. Here is a nitrogen with a positive charge. Here's another nitrogen with a positive charge. There's another nitrogen with a positive charge. Those are all basic amino acids. amino acids that have an oxygen in them, like this guy right here. And this one also has a nitrogen. It's polar, but it doesn't have a COOH or a COO minus, so it's not acidic, it's not basic, it's just polar. Now, one of the things that's going to be really frustrating to you if you try to do this too much without relying on a table, you're going to really quickly find some inconsistencies in the way that these amino acids are classified. Like here's one right here. If you're just going by the little tree diagram, look at that thing right there. That should be a basic amino acid, but it's not. Now, I'll tell you that if you are looking at a different data table somewhere else, a different data table may very well classify this amino acid as being basic. So there's a lot of disagreement on how these amino acids should be classified. Most of them, everybody is in agreement on, but there are going to be some things that you'll see that will vary from one data table to another. And we all have to just be okay with that. All of the problems that you have on sapling, they've been selected to avoid those amino acids that nobody can seem to quite agree upon. So it's not going to have, it's not going to have a big effect on how you do in the class or on your assignments.